welcome all of you. I hope this morning was not too tough, but it was really interesting. And what I'd like to talk today about is really what I've been doing in Geneva Hospital. So it's more of a kind of information for people who would like to embrace the career of a clinical bioinformatician. So it's mostly 3 parts. So the first one is about how we do software development in the clinics, research in the research lab. Then I'll go on with a few daily routine examples, what most bioinformatician, clinical bioinformatician will do. And third, which is, to my opinion, the most interesting one, is the potential for innovation. So, clinics versus research. I don't have to tell you that this is different. It's a different context. Valerie also mentioned it a lot. Different problems, different regulations, implications. We need the high precision, high sensitivity. For instance, we don't want to miss a treatment option because everything is linked to the patient in the end. So if we fail to report something, like for instance the mutation, then we might miss a treatment option for the patient. And in the same way, we don't want to give false results because there are high stakes for the patient. For instance, if we detect a BRCA mutation, and actually that was wrong, there was no BRCA mutation, the patient might undergo a double mastectomy. And this is really drastic if we made an error here. We can't come back 2 months later and say, well, actually, sorry, my bad. You didn't need to remove both of your breasts. So we need high precision, high sensitivity, and we need high reliability as well, because analyses are performed weekly. And the results are usually wanted for the next day, if not the day before. So you don't, as a bioinformatician, you don't have time to say, well, okay, yeah, I know there's a bug. I'll come back, I'll fix it and come back to you next month. It's going to be way too late. The doctor wants the results now. Yeah, there we go. Now, Valerie already talked a bit about the differences and mentioned the regulations. This is something that really struck me because I was in the research lab before and I moved into the hospital. And what one has to know is that laboratories in the hospital are accredited. So they have to follow a norm, it's the ISO 15189. And on top of that, you have internal IT regulations. So usually hospitals have very tight IT security because you don't want to leak data to the outside world. It's patient data, it's highly sensitive, or they don't want to lose data as well. And something that you might be confronted to is, can you actually send data, clinical data, into a cloud server in the US or in a cloud server you don't know where? And that's something that's really important to account for. And last but not least, there is this law called the medical device law. It's the ODM in French. In Switzerland, Ordonnance Dispositif Médicaux. I don't know in German how it's called. And in European Union, there's a new law that came into effect in 2017. So what is a medical device? A medical device is something that has a potential impact on the health of an individual. So for instance, if you take a needle, that's a medical device. But a software can also be a medical device. For instance, if you use a software to determine the 21 trisomy on the prenatal, on the fetus, that has a high stake. So you want to make sure this one is really doing what it should be doing. Or if you use a smartphone app to calculate the insulin dose, or if you use, and well, I'll talk more about this, an NGS variant caller to detect mutation. Now, if you're lucky to be in a public health institution, the regulation is a bit much lighter. So in Switzerland, there's parts of the whole ordinance that don't apply, but you still have to apply most of it. And for the new EU law, you only need to respect the Annex 1. So just to give you a small overview, this is Annex 1. It's 12 pages of low text. It's terrible. And if you want to respect all that, you're going to spend half a year just documenting your whole software. It's really troublesome for a small team of bioinformaticians. Now, this is the regulation that you need to follow for research-grade software. It's not a mistake, it's empty. There's no. There might be actually some journals that says, Well, if you want to publish in our journal, your code needs to be open source, needs to follow the good practices, be documented, and so on. But there's no truly regulation for research grade software. And that's the big, I would say that's the big difference between developing software for the clinics versus developing for research. Now, let's move into a few examples of what I've been doing, and I think most clinical bioinformaticians are doing. NGS variant coding. So the situation is that the lab has a new NGS panel or a new sequencer, and they need an analysis pipeline. So they go to you and say, can you please do something? On a small apartheid, there was this nice picture I got on the Illumina website where you see a lab technician with an iPad. So if you're working in a hospital, you're not going to get an iPad. I'm sorry, guys. And I still don't know what this technician is doing with this iPad, because either they're really, really advanced and they have the ability to communicate from the iPad to the sequencer, but most likely, if you would see that now, most likely the technician is working the soccer game. Now, so we have to pick to make an analysis pipeline. So how do we do that? We have to pick a software. So first question we need to be asking is, who will be using the software? Is it going to be a bioinformatician? Is it going to be a lab technician? And that changes drastically, because if it's a bioinformatician, you can use command line. If it's a lab technician, you need the graphical interface, and that's something on another scale. So you could buy a software. That's going to be way easier to maintain. You don't need to develop it at all. You have support, maybe not 24/7, but you have some support and so on. The downside is that you might have some potential compatibility issues with the other software in the lab. And very often, if the software is something that relies on computation, like NGS variant calling, it relies on cloud computing. So you have to send data to the cloud. And for that, it's going to be really complicated with the internal IT regulation. Some vendors will sell you the machine, but usually it's really expensive. And on top of that, as we've seen, if it's a medical device like NGS variant caller for a rare condition or prenatal trisomy, then it should have a CE-IVD tag. And if you have this tag, the price goes up because they have to do extra work on their side. Now, the other option is to develop in-house if you have a bioinformatician. So you can use open source tools, and this is usually, you have very powerful tools that are open source. The issue with open source is that you never know if it's well maintained, if it's been well tested, if it's well documented. So you have to spend a lot of time to make sure the tools you're using are going to be really reliable. And in the end, if you have a bioinformatician that spends roughly 6 months developing a software, that will cost you like roughly 50,000 francs. So that might be cheaper to actually buy the commercial software. But the advantage from having in-house development is that it's going to be perfectly tailored to the task. So you can tweak the data, you can tweak the algorithm, you can make small changes all the time whenever it's needed. And that's something that is really interesting for a lab in a hospital. Other small things that you might do and that I've been doing in my daily routine is installing and validating new software, especially if it's highly specialist software. I'm not talking about installing a new email application. I'm installing new NGS related software and so on. Helping the lab with the validation or the choice of a new NGS panel. Usually the clinical bioinformatician has this expertise on the tools and the genetics behind it. It's interdisciplinary. So very often we participate in the clinical reports and the tumor boards. And last but not least, if you're making your own application, you're going to spend a lot of time debugging and also explaining to the users why it's not doing exactly what they were expecting it to do. So user support is also quite important. You have to communicate a lot with the people in the lab and the clinicians. Now, one last point about the daily routine, which I think might be actually the most important, is bridging the gaps. In the clinics, you have you never have like one single software that does everything from the beginning to the clinical reports. There's always like 3 or 4 softwares, and they need to communicate, and very often it's not automatic. So people will need, for instance, to download the results from the variant calling software and upload them into the hospital system. Or even worse, they might need to manually recopy the result. And this is a high risk of error. Now, if you're a bioinformatician, you can automate this task, or at least try to simplify them so that there's at least no manual recopy. And with that, you're going to gain time, you're going to gain security, and so on. And this is one of the big projects we did in collaboration with the SIB in Geneva. So talking about NGS workflow, we had in the beginning, this was the workflow in the beginning. Everything that was before the sequencing was done with the lab notes, paper, Excels. And then they would copy, I think manually, they would recopy manually on the sequencing platform, on the sequencer, all the information with the barcodes, the lab, the ID of each samples. Then they had to launch manually the variant calling, and then they had to download the result, upload them in a search software to validate them, and then they had to copy again into the hospital system. So we developed a new tool called OncoBench, which tries to really bridge the gaps between all of these softwares and remove all of the lab notes on paper and Excel sheets. So in the beginning, it was really focused on somatic mutation detection for oncology. Now it's being used, at least the first part is being used for germline mutation, and we added copy number as well. The biggest point is really that we replaced all the paper steps and the Excel steps into one database. So everything is tracked, everything is secured into a hard drive that is in the hospital system. There's no possibility to lose data and so on. And also it's a web-based, so there's not one PC that has the software. It's available for any PC from anywhere from the hospital. So if you're in a room, in a meeting room, you can still access the software. Now the vision for this software is really to grow and to be the central software for high throughput analysis, at least in Geneva. This is how it looks. So the idea really to have a really neat interface, really simple, and going from the lab needs on what they were doing actually, looking at what they needed as information to annotate to do their job, and try to display that in the best possible way. A few words about the product. It's been like 5 years since we started. We release one new version every year with added support for new sequences or new panels and so on. What is really interesting is that after 3 months, we did a small user survey, and we realized that they were gaining 1 to 2 hours weekly for a pathologist and a biologist. So usually annotating the result takes at least 1 pathologist and 1 biologist. So it's roughly 4 hours gain of time that you would see using a software like this. And there was also other point like higher quality of results, better interpretation of variants, user-friendly, easy to learn, and so on. So that was one example of the daily routines. Now, I'll talk more about this potential for innovation, or as I might call it, side quest. The side quest is really something like someone coming to you and say, hey, I was wondering if you could help me with my code creation, because I had this idea that maybe this BRAF mutation is linked to the response to the treatment we've been giving for the last 5 years. Can you tell me how many BRAF samples we have in the hospital? I say, yeah, sure, I can look it up. Everything is in the database. Or maybe someone comes to you and say, can you explain, we got the result from the sequencing and there's like so many mutations. Why is that? And one possible answer is that, yes, we can compute mutational signatures and we can tell whether it's likely MSI, tobacco-related, UV, and so on. Another query I had was, is it possible to compute the oncotype DX from the fusion panel? So the fusion panel is a technology we have. It's RNA-seq, and it's aimed to detect fusion between two genes. So I could have computed the oncotype DX if the panel had contained the right set of genes, but the panel only contains genes that are implicated in fusion, and this is not the same genes that I used for the oncotype. So no, I can't. And plus, I'm not sure I should, because the oncotype DX is patented, and probably I can't just copy it and use it. Now, one side quest that is really interesting, and maybe that's because of the latest one, is about HRD. So there's this guy coming to me and say, hey, I was wondering if you could compute this HRD score that my colleagues keeps talking about. So what is HRD? In the beginning, you learn a lot during this job. So every time someone comes to you, you're always learning something. So what is HRD? HRD stands for homologous recombination deficiency. So homologous recombination, that's the pathway that handles DNA double-strand breaks repair. And it's most well known for BRCA1 and 2. So why the hype? Why the hype is because it's a pan-cancer mechanism. It's frequent in triple-negative breast cancer, high-grade serous ovarian carcinoma, and prostate carcinoma, roughly 50% of the cases. But that's not all. The main point is that it's actionable. And that's really the crux of clinical bioinformatics. Everything we do to be of interest for the clinics has to be actionable. They need to be able to do something with it. And the HRD is something that is actionable through synthetic lethality via PARP inhibitors. So at the bottom right, you see a small schema of how the PARP inhibitors work. So if you have DNA damage, you have a single-strand break. And usually the single-strand break is repaired via PARP. And if you have PARP, then the single-strand break is repaired, and this is fine. If you put a PARP inhibitor, you cannot repair the single-strand break, so you have double-strand breaks. And the double-strand breaks, they are repaired by the HR, the homologous recombination pathway. So now if you have a cell that have an HR pathway that is effective, it can repair, and it's living fine. So a normal cell will be living fine with this, with a PARP inhibitor. But the cancer cell that has a mutation in BRCA or the HR pathway will actually die because it cannot repair the double-strand break. Now, if we want to detect this deficiency in the HR pathway, we have two ways. The first one is to look for the cause, so look for the mutation, like a mutation in BRCA, or we can look for the effect. We can see if there are indeed many double-strand breaks. And there was one study, there's been a few more clinical trials, but there's one clinical trial with a PARP inhibitor called Paula-1, where they see that they had roughly 30% of patients that had a mutation in BRCA, but they had also 20% where they could see a phenotype of BRCA-like mutation. So that means, and this 20% actually also responded to the PARP inhibitor. So there is added value to look further beyond the BRCA mutation. And if you test only for the BRCA mutation, you miss 40% of patients that could actually respond to the treatment. And that's a lot. Now there's been a commercial test available by Myriad. And what they show, it's published data, that they have mostly 3 patterns they could identify that are linked to BRCA loss. So the first one is telomeric allelic imbalance. So at the end of the chromosome, you have duplication of the end of the chromosome or loss of the end of the chromosome. Or you have loss of heterozygosity. So you lose either the maternal allele or the paternal allele. Or you have what they call large-scale state transition, where you have large segment, more than 10 megabases that are duplicated or lost. And based on these 3 markers, they make the test. Unfortunately, when we started the project, I think it was 2 years ago, the test was not available in Europe. So it was available for clinicians in the US, but not in Europe. And plus, it's really expensive. It's roughly 6,000 francs. to get the test. So we had this idea to develop the test in our lab. We already had a gene panel to cover the mutations, so that was not an issue. But as we have seen, if you just cover the mutation, you're gonna miss a lot of patients that could respond to the drug. And we had in the lab this OncoScan assay from Thermo Fisher Affymetrix that we used to detect copy number alteration of the whole genome. And it was mostly used to detect losses of chromosome arms and to detect focal gene amplification or deletion. But this technology actually gives us all the information we need to compute a score similar to Myriad. And we could actually redo exactly the same score as Myriad, but we can't because it's patented. And if we were doing a research project, that would be fine. We could just apply the Myriad score. That would make publicity for Myriad. They would be really happy. But there we're trying to make a score to treat patient. So we basically on the same business as they are. So we can't use the same score. So what we had to do is develop an alternative score, which is very similar to Myriad. The point was to have something that is similar to the gold standard, which is the score by Myriad. And this is something we've done. It's been in routine since a year. And since we have a lot of available data in public cohorts like the TCGA cohort, so the TCGA cohort, there's a lot of cancers, but there is 450 ovarian cancer and 100 triple negative breast cancer. And if we compare the score from Myriad that we could compute, because they tell us how to compute it, and the new score we see, we have a really nice agreement between the two. And that would be mostly it for today. So if you had to remember 3 things, I hope it's not too much, is that clinics is not equal to research. We have different quality requirements, different regulation and implication. A second thing would be that clinical bioinformatics is really, in my opinion, it's often there to bridge the gaps between the different software in the labs. This is what you're going to do most of the time. And also what can happen is that the clinical bioinformatician will really translate a research software into clinical grade. We have a lot of samples in the labs and samples that have been previously studied, characterized. So there's a gold mine of information. And if you need to validate a research software, you can do that in the lab and make sure it's reliable, it's doing exactly what we need. Maybe you need to combine it with another software, add a few filtering steps and so on, change the settings, but that you can do with respect to all the samples you have. And last but not least, is the potential for innovation. As we've seen with, for instance, this HD score, we really have a potential to develop new tools and computer science tools without having to develop new techniques or buy a new panel or buy a new sequencer and so on. So I think this is really the most interesting thing that a clinical bioinformatician can do in a hospital. And with that, I'd like to thank you for your attention, and I'll be glad to take any questions.
Thanks a lot, Yann. That was a really great talk. I think it really highlights, I guess, the daily and routine perspective of a bioinformatician, which is like actually very different from research, and the monumental task of, bridging all the different softwares together to generate a pipeline. And then also what's really nice is you can then develop new pipelines based on research questions that people ask you, and then implement them in routine. So we have a comment here where they said that if you want to use off-the-shelf software as opposed to custom-made, you would spend 6 months upfront defining the requirements, evaluating offers, and so the overhead would just go to project management. And then also in the same case, you would basically need someone who knows enough about the software to kind of evaluate if it's suitable for the application. Do you have like an opinion on this?
Yeah, so obviously picking the right software is important. And if you buy off-the-shelf software, and this is something we do quite often, you have to know what you're going to buy. So you're going to spend some time. It's not going to be the cost of buying this software is not going to be just buying the software. You're going to spend some time But I still saying that defining the requirements, defining what you want, you're gonna have to do that as well if you want to build, make a custom self-made software. So this part is not going to change, but the development part is going to change.
Okay, we just have one question here, and then I will get to Daniel after. So the question from Q&A, they asked, if I understood correctly, the HRD score at HOOG was validated in silico with TCGA data. Did you also perform clinical validation using in-house samples?
So yes, there was, I presented it very lightly, very shortly. We validated on samples that we run ourselves. We have roughly 100 samples that have been tested with Myriad, and we could compare to it. But this data is still private, so I'm not allowed to disclose it yet. But the result was really nice. It's really concordant with the Myriad test. And we're currently running 300 samples from the Paola study to see if the score is really predicting the clinical response to PARP inhibitors. So we're going to have the clinical validation of the test by the end of the year, most likely.
And Daniel, you can ask your question.
Sure, thank you. Great talk, Yann. So you somehow emulated that Myriad score and you got it into the clinic. I assume with my limited knowledge that you had to certify this process to actually use it in routine diagnostics. Now, if you tune one of these parameters based on your research findings or by improving the score, will that end up in a recertification? Or is there, can you briefly comment on how that would happen, or is that not happening?
So we didn't use the Myriad score in routine because that we can't. There's a patent on it. So the whole point of re-implementing the Myriad score was to really being able to compare it on our own data. So that was the main point. And then for the certification, what you need to do, since it's a lab test, internally developed and for internal use, it's a bit lighter. So you don't need to have it accredited per se. You just need to follow the internal regulation for validation of a new test. So you need to show that you made the validation. You need to show that the documentation is on par, that the security of the system is right, and so on. But you don't need to have it accredited officially.
Thanks.
Okay, I think we have no more questions. If you have any questions you think of, you can also bring it up in a panel discussion after our next talk. And so we will move on. Thanks, Yann, again for your great presentation.